Well, hello, ladies and gents. Hope you're all doing well today and enjoying the day. So, in this lecture series, we're going to begin to look at the life cycle of cells because since all life is made of cells, the fact that life continues is based on the capacity for cells to basically divide or make new copies of cells. So, it's been this sort of spectacular process that's gone on for at least 3.5 billion years where cells create new cells. And so the cells that make up you are descended from cells that you inherited from parents and on back in time. So it's sort of this remarkable and spectacular continuity of life that's existed over time. And now you're lucky enough to possess some of that sort of inheritance of life. So in this chapter, we're going to look at how that happens that cells can go about dividing. Okay, so the cell cycle refers to the entire lifespan of the cell. Cells actually spend most of their time not dividing. They're doing their sort of cellular work, right? But cell division does comprise an important component to the cell's lifespan. So let's sort of jump into looking at why cells might divide. Again, as you sit here, you're made of trillions of cells, but those cells are descended from one cell, the zygote, that began your existence. So to grow and develop, you got to do that by adding cells. Repair also tends to happen with tissues over time. Think of like if you've ever had a cut, that cut heals because cell division has been prompted there, and cells replace those cells that were lost. Another important factor that we don't have on here is efficiency, right? Think of like the little Indy, big Indy example. So as cells get larger, volume increases faster than surface area. So cells get bigger and bigger and bigger. Surface area to volume ratio goes down, 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 down. And if cells become less efficient, they can't get rid of waste and take in raw materials fast enough. So efficiency is another reason for cells to divide. All right. Now, it's important to distinguish between mitosis and meiosis at this point. Not all the time, but frequently on the AP exam, they'll ask a question sort of comparing mitosis to meiosis to see if people trip up on that. These are fundamentally different processes done for absolutely different purposes. The purpose of mitosis is to create two genetically identical diploid daughter cells. So you're basically making new copies of cells. You're replacing stuff or adding to. Meiosis, you create four haploid unique daughter cells. The purpose of meiosis is to make the gametes, egg and sperm. So meiosis is a niche process. Now, some of the steps bear similarity. You know, they have sort of similar mechanisms occurring, but they are distinct processes done for different purposes. So no differences between mitosis and meiosis because sometimes that stuff creeps up. All right, now let's contrast genomes with garden gnomes. The purpose of garden gnomes is to startle dogs and small children. Genome is in reference to the complement of DNA that's found either within an individual or within a species. So you can speak of like the human genome. So with the human genome, we're looking, what are the 25,000 genes that the human species possesses? Or you could look at individual genomes. So of those 25,000 genes, what copies do I have or do you have? So you can get your genome sequenced for, I think it's under $1,000 now, but you can figure out what copies of all these 25,000 genes do I have. Again, a genome is in reference to all the genetic material. Now, DNA can come in a couple different varieties. There is the DNA that you find in prokaryotes, like bacteria. They have one loop or circular ring of DNA. Or you can have DNA in an arrangement like what we have as eukaryotic organisms. They're linear chromosomes. They're lines or threads. Okay. Now, DNA is always DNA, right? It's a double-stranded molecule with A's, T's, C's, and G's, and it stores code. Now, DNA can sort of change its shape. And when it changes its shape, we refer to it in slightly different ways. DNA, when it's all coiled up and getting ready to divide, is called a chromosome. I like this image because you can see sort of the untwisted ladder of DNA. You have the nitrogenous bases in the middle, and then there's sort of a generic sugar phosphate backbone along the outside. And then it twists into a double helix. And then DNA coils up around these histone proteins. It coils up really tightly. And then it can form these loops and whorls and fold back in on itself into a chromosome. Okay. Now, when DNA is all tightly coiled up and packaged up into little threads, we refer to those as chromosomes. The prefix mitos means thread. So when we talk about mitosis, you're talking about the DNA all coiled up into chromosomes. So under the microscope, it looks like little threads in the nucleus. During interphase, the period between cell divisions where cells spend most of their time, DNA is in a relatively relaxed state, and a lot of times we refer to that as chromatin. So DNA wrapped around histones is technically what a chromatin is, but a lot of times people talk about chromatin, it's DNA in a relatively relaxed state. And that's important because, like, if we look at this image here, you see the DNA wrapped around the histones, the intervening space between these histone proteins, that can be unwound relatively easily. Then you can access the code in there. So when the DNA is all really tightly coiled up into a chromosome, you're not getting at that code, you're not going to be making any proteins. So during interphase, when cells are sort of going about their cellular work, you need access to the DNA. So the DNA sort of relaxes so you can separate the strands relatively easily and then transcribe that code. So, water has all these different versions, right? You can have gaseous water, solid water, liquid water. DNA is always DNA. It's just sometimes really tightly coiled up and sometimes it's more relaxed. All right. We have a couple different types of cells we're going to look at here, somatic cells versus gametes. The prefix soma means body. So think of somatic cells as being like your body cells. These are all the different types of specialized cells that do all these different jobs throughout the body. So almost all the cells in your body are somatic cells. And somatic cells are diploid. You have two sets of chromosomes, one from biological mom, one from biological dad. And they're non-reproductive, so you don't make new people from somatic cells, with some notable exceptions. Okay, now the other type of cells we'll look at in this case are the gametes. The gametes are the sex cells, egg and sperm. These are haploid cells with unique combinations of chromosomes. So again, gametes are produced through meiosis. All the somatic cells, most of the body cells in your body, they're created through mitosis. All right, here we're going to look at sort of the structure of a chromosome. Before you separate all the DNA, now if we think about it, you've got, like, in a somatic cell, 46 chromosomes. Those 46 chromosomes, if you unwound them completely, like you unwound them from around the histone and put those 46 chromosomes end to end, they'd be this big. You have about 6 linear feet of DNA. So that's a lot of information. So if you imagine 6 literal feet in 46 pieces, that stuff can have molecules bang into each other and they can shred themselves pretty easily if you try to separate them. So before your DNA gets separated, it's going to coil up or condense into those chromosomes, so it's packaged up a little more tightly, so it's less likely to experience those breaks during separation or mitosis. So when we look at a chromosome, what will happen is during interphase, that DNA is going to be replicated. So that chromosome is actually made of 2 sister chromatids held together along their length by a protein called cohesin. So this cohesin protein sort of stitches together, holds together the 2 sister chromatids. Okay, so the image you see there is one chromosome made of 2 sister chromatids, referred to as a replicated chromosome. Okay, so a chromosome is made of 2 sister chromatids held together by that cohesin protein in the middle. Now, there is a characteristic region within a chromosome called the centromere. I just think the centromere is in the middle. It's sort of like a narrowed waist. So it's really just a demarcation or like a landmark within the chromosome. So there is a region in the chromosome, again, that's called the centromere, where chromosomes narrow. So the centromere will actually differ between chromosomes. Sometimes in chromosomes, the centromere is characteristically sort of in the midway. Sometimes it's more toward the top or bottom. I mean, top and bottom can flip, certainly. But, the centromere, the narrowed region, can differ in the area. So just remember, one chromosome is made of 2 chromatids. So 2 copies of that genetic information, 2 copies of that DNA. All right, we'll end on this slide. We're going to look at differences between mitosis and cytokinesis. Cell division, you got to be careful, like on a test. If they ask about cell division, mitosis, or cytokinesis, they could be referring to slightly different things. Cell division is literally one cell becoming 2. Mitosis, remember that mitose terms mean thread? Mitosis is concerned strictly with what's happening within the nucleus. Okay, so what's happening to the DNA? Cytokinesis, cyto means cell, kin means movement. So cytokinesis is focusing on the division of the cytoplasm, everything else other than the nucleus. So sometimes it can be confusing because these two events tend to, well, they co-occur. One happens while the other is occurring. So mitosis is this process where the nucleus is dividing. And at the same time, near the end of mitosis, you'll also have cytokinesis, the division of the cytoplasm. So keep those distinct in your mind. If they're talking in a prompt about cell division, they're talking about both mitosis and cytokinesis. If they're talking about cytokinesis, they're talking just about the division of the cytoplasm. And if they prompt you about mitosis, they want to know just what's going on in the nucleus with that DNA. All right. So yeah, we'll stop there. Thank you much.